Okay, we are on air and we have lots of attendees joining us. Amazing. Hi, everybody. Thanks for joining us today. This is our first time using Zoom. We've recently switched our webinar software because of the amazing interest in this work. I'm super excited. I know a bunch of people are still logging on right now. My name, for those who haven't joined us yet, is Brad Burge. I'm the Director of Strategic Communications for the nonprofit Multidisciplinary Association for Psychedelic Studies. And we are just incredibly excited for this webinar series. We're now starting our second in our series of 7 of the Psychedelic Science webinar series. Today, we're going to be talking about psychedelics and the brain with Dr. Katrin Preller joining us from Switzerland, hosted, of course, by my amazing colleague, Dr. Bia Labate. I will be signing off in just a moment on my video, but I'll be available in the attendee chat. I just want to give everybody a quick rundown on what Zoom is like. So first of all, we've been amazingly successful with this webinar series so far. We had this planned over 7 months ago. Did not know that we would all be sheltering at home and that all of the in-person psychedelic events would be canceled. So thank you so much for joining us. We have over 1,400 people signing up for this webinar series. Encourage you to participate in the attendee chat. You can access that down at the bottom of your screen. There's so many of us here, won't be able to read everything, but I'll be putting in some comments and links and answering some questions there. If you do have questions or need any kind of technical support, you can't get in or anything like that, first off, try closing Zoom, opening it back up with the same link. That solves about 99% of problems. If you have more problems, you can email us at webinars@maps.org. I'll put that in the chat as well. So this is going to be recorded. For those who have paid for registration for this, you will also receive access to this whole session recording, including the slides, after the series is over. So I suggest also that you close any other programs running on your computer to avoid any sort of overlap. Last but not least, for the question and answer period, which will happen after Dr. Preller's presentation, enter your questions in the Q&A box down at the bottom of your screen. If you do put a question in the chat, there's no guarantee we're going to see it. So if you put it in that Q&A box, we'll be looking through those and sorting them and combining them so that we can answer as many of those questions as we can. So yeah, a lot of people are already doing it, but please share where you're coming in from in the chat. We love to see the diversity of folks. We have over 600 people now here, and I'm super excited today to hand this over to Bia. Thank you so much, Bia, joining us from San Francisco. I'm also in the East San Francisco Bay Area, and I'll let you two take it from here. Thank you so much.
Thanks, Brad. Hi, everybody. So my name is Bia Labate, and I have a PhD in social anthropology from the State University of Campinas. And I work at MAPS, where I'm public education and cultural specialist and host of this series. We're very happy to have a brainy, nerdy subject today led by a woman. So that's especially rare. Always when we put together these events, we're trying to look for parity in gender and also racial diversity, and it's so hard to find women in science, and it's very special to have only women. I just want to remind everybody that originally Dr. Stevens Rain, who's a good friend and colleague from Brazil, he was supposed to be sharing the space of the webinar with Katrin, but his lab was called to work on coronavirus related issues in Brazil. The situation in Brazil is pretty bad. And he's a very top high-end scientist, and he's collaborating on doing research and providing support. So he sends his apologies. He won't be able to join us, but we're very happy that he's doing this important work, and also thank Katrin in advance for agreeing to be the single presenter and putting this extra burden on her, and reminding that she's also donating her time for us, and we're not paying her, she's volunteering, so that's really important. We're grateful to be able to invest in psychedelic education and research. So I already told people the last time a little bit the general objectives of the webinar. We want to bring academic knowledge to a more accessible language, make it wider so lay people can understand. We want to be able to give people a chance to interact with researchers and clinicians and learn about the backstage of their research and aspects that normally don't appear in psychedelic science conferences or in peer-reviewed journals. We also want to show the public that doing research is actually kind of fun and inspire new generations to do research. And some people feel afraid because of the stigma related to psychedelics, and they're doubtful whether they'll take this research path. So we want to stimulate the field to grow, to be stronger, to connect directly to the researchers. And also on the other way around, to give the researchers an opportunity to listen from the community. What are some of the worries and some of the things that people wonder, and in which directions they think research should be moving to. And today we are going to talk about psychedelics and the brain. For me, as I was making a joke with them, that for me as an anthropologist, that is the truly hallucinogenic part of things, because I find it abstract and hard to understand all the science. But I'm happy that Catherine is giving a lecture that I hope will be accessible to all. So she can talk more about her background, but just a little intro. She has a PhD. Her PhD is from Constance in Germany, and now she's based in Zurich. And she has been working with psychedelics in the last 10 years, mainly in the university. Her PhD is from Constance, but she is doing work in Zurich, and now she has also affiliate professorship position in Yale. So she will talk about how the experiences, in a general line, how does these experiences work in the brain? Why and how psychedelics work in the brain? And one might ask, why do I care? Why is that important? Well, that is important because a lot of diseases generate in the brain. So there's a lot of potential to understand how to treat new diseases. There's also a big discussion on deficits on social cognition. She will also be addressing these issues, and also how maybe studying psychedelics can bring light for us to understanding aspects of the brain as well in itself. So with all those words, I will pass the speaking slot to her. And as Brad said, please post your questions and answers, and we'll talk later in the quick questions and answers. Thank you, and Catherine.
Yeah, Bia, thank you so much for this nice introduction, and I'm so happy and excited to be able to talk to you today. Yeah, as Bia just said, I'm a researcher mainly in Zurich, also affiliated with Yale, and my talk today will be about the, what exactly do psychedelics do in the brain? And as Bia said, I hope that I can encourage you to think about the science of psychedelics as fun. Well, let's see. Let's see about that. So I'm going to share my screen now so that you can all see my presentation. So just give me a couple of seconds for that. Okay, so I'm not necessarily able to see you anymore. I hope that that it's working. Just another quick second. Okay, here we are. Yes, working. So as I said, I'm going to talk about the effects that psychedelics have on the human brain. The two psychedelics I'm going to talk about are psilocybin and LSD. So they have pretty similar effects. However, their pharmacology is pretty different. So psilocybin is a serotonin 2A and 1A receptor agonist, and LSD stimulates a lot of serotonin as well as dopamine receptors. So I'm trying to make this a little bit clearer by the next slide here. So what does it mean to be a serotonin agonist? So what you can see here is a neuron, and specifically it's a synapse. So this is how the brain communicates, and usually it communicates by releasing neurotransmitters, which then act upon the following receptors. And as I just said, most of the psychedelics act on the serotonin 2A receptor. So down here you can see the serotonin 2A receptor. Now, what psychedelics do is, instead of using the brain's own neurotransmitter, it docks to the serotonin 2A receptor and releases a full cascade of things that then happen in the cell, in the neuron. So this is a little bit different from what, for example, MDMA is doing, because MDMA is mainly releasing the serotonin that the brain already has. However, psychedelics are replacing basically the serotonin. They're acting as serotonin, however, very specifically on mostly the serotonin 2A receptor, and in the case of psilocybin, also the serotonin 1A receptor. And what happens afterwards, so don't necessarily look at all what's going on here. So in short terms, that means that if it docks to the serotonin 2A receptor, it's more likely that the neuron will fire more strongly. And if it docks to the serotonin 1A receptor, the neuron will rather stay silent. Now, talking very briefly about the subjective effects. So both of the substances, on the left side, you can see LSD, on the right side, you can see psilocybin. Both of them induce a very specific altered state of consciousness. So they're both psychedelics. So what we usually see in our studies is experience of unity. Some people experience this as spiritual, a blissful state or insightfulness, changes in how we perceive our body, impaired control and cognition. Also, some persons experience some anxiety, as we can see here. Luckily, in our studies, we try to create an environment where people do not necessarily get anxious. And then, of course, all the visual alterations that people experience, and that gave rise to the name hallucinogens. As I said before, psilocybin and LSD are pretty similar in that way. However, the duration of action is pretty different. So when we administer psilocybin to our participants, it's usually, they usually have this experience for about 6 hours after we administer the substance. After that, they feel pretty sober and usually can go home after that. With LSD, it's a bit different. There, depending on the dose, the effects last a lot longer, usually up to 10 hours, depending on dose. It can be up to 24 hours. So you can already imagine that doing research with LSD is much more complicated than with psilocybin. And that is probably also one of the main reasons why psilocybin is used more for therapeutic purposes right now. So talking about therapeutic purposes, there has been an increasing amount of studies looking at the clinical effects of psychedelics. And we're currently running 2 studies in Zurich as well, one where we're looking at psilocybin for the treatment of depression, and one where we're looking at psilocybin for the treatment of alcohol use disorder. However, I'm not going to talk too much about these studies today. Rather, what I would like to talk to you about is our studies that we have conducted with these substances in healthy human participants. And the reason why we do these studies in healthy participants is, first of all, that these substances offer this unique opportunity to investigate things that we usually cannot investigate, or at least not easily, in healthy participants. So we're talking, for example, about changes in body perception. They're really hard to induce non-pharmacologically. However, with these substances, we can induce them reliably and therefore study them. On the other hand, we also believe that by increasing our understanding of how these substances work in the brain, we can hopefully help to uncover their full clinical potential. And I'm going to show you a couple of examples of that. So as Bia said in the beginning, I will mainly try to answer two questions today. The first one is, what are the effects of psychedelics on the human brain? And the other thing is, how do psychedelics influence how we process social information? So in the first part of the talk, I'm going to talk a lot about brain connectivity. So you'll see a lot of pictures basically showing fMRI data of brain. So basically what we do is we administer the substance and then we put our participants in an MRI scanner and well, look at their brains. And most of our subjects, basically all of them, really tolerate this really well. So surprisingly, they find it quite comfortable lying in the scanner and tripping. But before we go into too much detail, I want to take a brief look at the brain itself. So what you can see here is our colored areas of the brain, of the human brain, and gray areas. And there is an important distinction between these colored areas and the non-colored areas. So the colored areas are basically the areas that process our sensory information. So in these pink areas at the back of our brain, this is where our visual information is processed. Here's where our auditory information is processed. Here's where our touch is processed. And then we have all the gray shaded areas here. And these areas are responsible for integrating all this information that is coming from these sensory parts of the brain and make a coherent picture out of this information that they get, and then use this information to, for example, plan the next steps that we want to do. So to repeat briefly, we have these sensory parts of the brain, and on the other hand, we have these association regions, which are responsible for integrating the information and basically representing the reality as we usually experience it. Now, there have been and still are quite a lot of hypotheses how exactly psychedelics change the way our brain works and how our brain communicates. So we've seen these different areas, and of course they are communicating with each other, because otherwise we would not experience the reality as coherent. So one of the theories that was suggested almost 20 years ago is that we have a certain part of our brain, which is the thalamus in the middle here. And basically we receive all of the information from within our body or outside our body through the brainstem, and all of this information is led to the thalamus. And now the thalamus basically decides what information is important and distributes it to the brain. And it has been proposed that under the influence of psychedelics, this filtering mechanism in the middle of the brain here does just not work as well as it usually does, but it kind of unfilteredly leads the information to the cortex, which then leads to a sensory overload, because the non-important information is not suppressed. So the brain is overloaded with information, and this is then experienced as psychedelic symptoms. Now, what we wanted to do is we wanted to test this hypothesis empirically. And we did this under the influence of LSD. And the first way we looked at it is using a method called functional connectivity. So basically what we did here is we took the thalamus and then we looked at how it is connected to the other parts of the brain. And this is basically a correlation between the signal in the thalamus and the signal in the rest of the brain. Now, what you can see here, and now keep in mind the picture I showed in the beginning, this differentiation between sensory regions and associative regions, is a pretty interesting differentiation between these sensory regions, which are highly connected with the thalamus, and these more associative regions, which are loosely connected to the thalamus. So that, first of all, gave us an impression that it seems like indeed the thalamus is more highly connected to especially the sensory parts of our brain. However, this method has a major downside because it doesn't tell us anything about directionality. That means that while we here see that the thalamus is more strongly connected to, for example, the visual cortex, that doesn't mean that the thalamus is sending more information to the visual cortex. It could also be the other way around, that the visual cortex is sending more information to the thalamus. So to be able to overcome this issue, we had to use a different method. And this different method is able to give us actually directionality between brain regions. However, every method comes with downsides. So with this method, at least for the time being, we are not able to look at the whole brain, but we just have to look at certain parts of the brain. We have to choose regions, which we call regions of interest. So we did that, we chose specific regions in the brain and we ran the analysis. Of course, the thalamus is at the center here. And what we see is that indeed the thalamus is connected more strongly to certain parts of our cortex. In this case, it's the posterior cingulate cortex. This is one of the regions we tested. But also what we've seen is that it's not just undifferentially sending information to the cortex, but it's sending information to specific parts of the cortex, and it's sending information and it's sending less information to other parts of the cortex, which kind of makes sense because usually people do not necessarily just experience sensory flooding, but they have very specific symptoms. So it's not like all the information just goes straight to the cortex, but it's really region specific. Now, we've talked a lot about the thalamus. We've not really talked about what exactly is happening in the cortex then once the information reaches the cortex. So again, keep this picture in mind, sensory regions versus more associative regions. And this is what we saw when we then looked at the whole brain again. This is psilocybin, this is LSD. You can clearly see a very similar pattern here. And I'm going to show you these results unthresholded. So we basically get, these are the areas which are what we call significantly altered, but I'm going to show you an unthresholded image so you can actually really see the whole, what's happening in the whole brain. And if you keep this picture that I showed before in mind, you can clearly see this differentiation here too. So we have higher connectivity between sensory brain regions, but on the other hand, these more associative brain regions, which are supposed to integrate the information, they are more loosely connected. So what does that mean? Yeah, sorry, I'm going back to this slide. What does that mean? That first of all means that a lot of sensory information is being processed, but on the other hand, this sensory information can probably not be integrated the same way as it does in our usual waking states, which is probably the reason why we experience the reality very differently under the influence of psychedelics. Now, I think you've probably heard about brain networks, especially in the context of psychedelics. A lot of people talk a lot about the default mode network. And I'm showing you, it's a very busy slide, I'm aware of that. For those of you who are really interested, you can look up the paper. I'm trying to walk you really briefly through it. So the default mode is in panel A here, but obviously the default mode network is not the only network that we have in our cortex. And what I've been showing you before is that LSD as well as psilocybin, they do not necessarily affect just one of these functionally defined networks. They really work in the whole brain, which kind of makes sense because the serotonin receptors that we've been looking at in the beginning of the talk, they're really distributed in the whole cortex. In some areas a little bit more than in others, but you can find them everywhere across the whole brain. And what we see in our data is that if we want to look at these different brain networks, we indeed see that the default mode network is connected more loosely to the rest of the brain. So we see this disintegration in the default mode network. And the way you can see that here is basically, this is the green bars are placebo, the pink bars are LSD. Forget about the yellow bars for now. And you see this decreased connectivity in the default mode network, but not only there. So we have the frontoparietal network, and you see this one is pretty much affected too, as well as almost all other networks in the brain. And you can also see that, for example, in the visual network, we looked at that before, connectivity is increased. So it is really, these are really substances that affect the whole brain. Now, I briefly kind of want to picture this a little bit more schematically to summarize what we've learned from these studies that I just showed. So we've seen that information reaches the thalamus, and then the thalamus basically filters and distributes this information. And what we've seen is that it's not just sending information everywhere. We've seen it sending information to specific parts of the cortex, and some of them are the sensory regions, and it's sending less information to the associative regions. And here we see that there is a disbalance between increased connectivity in sensory regions that is not counterbalanced by our associative regions to really integrate this information. And this disbalance probably gives rise to psychedelic experiences like the visual symptoms we see here. On the other hand, this may be beneficial, especially for people who suffer from thinking patterns that are not really good for them. So, for example, in depression, we're usually seeing ruminative thinking patterns. So people are stuck in negative patterns, always thinking about how bad their life is, how bad they are themselves. So if we now think that the information that we receive from within ourselves as well as from the environment all of a sudden gets integrated differently, that might actually help to pull people out of these ruminative, rigid thinking patterns and open up new ways of how to perceive the reality and to perceive ourselves. So I want to share another few recently published results that I hope will interest you. We've not only looked at brain connectivity during basically the peak effects or the strongest phase of the effects, we were also interested in, well, what's happening after I took the substance? And what is happening 20 minutes after we give psilocybin to our participants is that, first of all, we see increased connectivity in these regions which are responsible for our vision to be more strongly connected. And this really nicely overlaps with what our participants are usually telling us, because the first symptoms most of them are experiencing is that the floor starts moving or the wall starts moving. So this is nicely reflected in this pattern here. And then at a later point, this is 40 minutes, T2 is 40 minutes after administration, we see that this differentiation between more strongly connected sensory regions and less strongly connected associative regions is starting. And that is usually really when also the true psychedelic symptoms are coming up. And at T3, which is 70 minutes after administration, you basically see the full-blown psychedelic pattern of brain connectivity. Now, we were also interested in what does this mean for a clinical population? And what we were interested here is mainly, can we predict from someone's baseline connectivity how strongly they will react to the substance? And again, here you see 3 scatter plots We cannot really predict anything for 20 minutes after administration, but it gets really better at 40 minutes. And then there's a pretty high correlation at 70 minutes after administration. So this is giving us a first clue that maybe looking at the people's baseline brain connectivity might already tell us how strongly they might react to the substance afterwards. And we've seen in clinical trials that the experience people are reporting afterwards has something to do with how well they feel after the treatment. And it would be really good if we had a measure to know beforehand who will really benefit from the treatment or not. And this still has to be tested in clinical populations, of course, but it's a first hint that the way our brain is organized might already tell us how we might react to the substance. So then usually when I talk about these things, I get a lot of questions about, well, so what about microdosing? Because so far I've only talked about real psychedelic doses of LSD and psilocybin. So this is work that has been completed in collaboration with Harriet DeWitt and her group at the University of Chicago. And they gave 13 micrograms of LSD to their participants and had them do a couple of tests and also put them in the scanner. And again, here is the brain connectivity. It's slightly different here. What we did here is basically the same as with the thalamus that I showed in the beginning. So we had one seed, one region we were interested in, and we looked how it is connected to the rest of the brain. So what we did here is we used the thalamus, of course, because that is one of the key regions that we were interested in, and we didn't see any changes. And that is not necessarily really surprising because as we've talked about before, is that the thalamus or the filtering, the changes in filtering of the thalamus is probably one part of what is really inducing these altered states of consciousness. And with 13 micrograms and with microdosing in general, you don't necessarily want to be in an altered state of consciousness. And it doesn't. And also when we ask people how they are feeling, they don't report any psychedelic symptoms at that dose. So we looked at another region which we thought was really interesting, and this is the amygdala. And the amygdala is basically our emotional center of the brain. And what we saw here is that there are indeed changes in the connectivity between the amygdala and certain parts of the brain. Especially there's a higher connectivity to these frontal regions. Again, the regions which are more the controlling the associative regions. And what we then looked at is if these changes in connectivity are associated with the mood that participants are reporting. And indeed, the connectivity between the amygdala and this dot here in the frontal cortex, here you can see it again. So the strength between this connection, amygdala to frontal cortex, was associated with a positive mood that people were reporting. Now, what does that, or what could that mean? Well, we've seen that a lot of people report microdosing to treat depressive symptoms, for example. We've also heard that some people microdose to increase, for example, their creativity or their work performance. When we do that in the lab, at least so far, we've not necessarily been able to replicate these anecdotal reports. However, when we look at these connectivity patterns, what might happen is that if someone is feeling kind of low, maybe showing first signs of depression, and LSD, a microdose of LSD is changing how the frontal cortex can control our emotional center, this might lift their mood. And then if someone who is not suffering from low mood or mild depression, for example, is absolutely conceivable that these people indeed benefit from it and can also perform better at work, for example. So this brings me to the next part of my presentation, where I'm mainly going to talk about social cognition and how these substances modulate social cognition. The reason why I think it is a very, very important topic to talk about is because alterations in how we perceive our social environment and how we interact with one another are core symptoms of many, many psychiatric disorders. And not only are they core symptoms, obviously the therapeutic process in general is also a social one. So if I have deficits in well, understanding social cues, for example, it is very likely that my therapist-patient relationship suffers from it, for example. But also it makes it really hard for these people to function. And by function, I mean, well, having a job, keeping a job, or even just finding an apartment. And a lot of people are severely suffering from this, and we currently cannot really treat this. So here we really wanted to increase our understanding of how social cognition works in the brain and how we can potentially modulate it. So first of all, what we knew from psychedelics is, of course, that they change how we perceive ourselves. And we thought, well, maybe changing how we perceive ourselves might lead to changes in how we perceive our partners, how we perceive others, and how we interact with them. So the question of this study was really, are these two things intertwined? And we used a paradigm where participants were lying in the scanner and they were interacting with someone outside of the scanner via eye movements. It was a pretty complicated setup. I'm not going to bore you with the details. I'm showing you the results right here. So first of all, what we saw in this paradigm is that indeed LSD is decreasing the differential activation that we usually see under placebo between this is me and this is the other. And at the same time, it is changing how we perceive this social interaction. So this gave us a first clue that the self and social cognition are indeed intertwined, and that changing how we perceive ourselves might also lead to changes in how we interact with others. Now, what do psychedelics do? How do they change social interaction? So I'm taking a tiny step back here and talking about a potential clinical effect of psilocybin. So we knew from previous studies that psychedelics decrease specifically how we process negative information. So when we show people negative pictures on a screen, we see that their emotional center, the amygdala, which I was talking before here, does not react as much as it usually does. But what might be much more important for our patients is really not looking at negative pictures, but looking at social rejection. We know that, we know that a lot of psychiatric patients experience a lot of social rejection. They usually have smaller social networks, and they have less people to rely on, which obviously makes their life harder. So we used a paradigm which is called cyberball. We did not invent that. There are quite a few studies published with that. And what happens here is that our participant comes to the lab. He meets two other people. In this case, it's Michael and Sarah. They are introduced to each other, and they are told that they will meet again in the scanner. And what happens in the scanner is that the participant can play a ball to either Michael and Sarah, and obviously those two can do the same. then what happens after a while is that Michael and Sarah start excluding our participant from this game. They just don't throw the ball to him or her anymore. And that might not necessarily sound like the worst social exclusion you've ever experienced. However, if you try it, it is really pretty frustrating. And a lot of people come out of the scanner and are like, wow, this was really, really mean. And the beauty of this paradigm is that it has been shown to reliably induce increased activation in a brain region called the anterior cingulate cortex. And therefore, this signal in the anterior cingulate cortex has also been called the social pain signal. Now, when we do the exact same paradigm in patient populations, now here it's an example of borderline personality disorder patients, we see that they react even more strongly to this social exclusion than our healthy participants. Now, what happens when we give people psilocybin? Well, first of all, they report that the social exclusion wasn't so bad for them than it usually is under placebo. Well, now you might be like, well, you know, they had psilocybin, they're probably totally in their own world. Maybe they didn't even know what was going on. So of course we try to control for all of that. So we asked them a lot of control questions, and they were perfectly aware that they were excluded from this game. It just didn't touch them as much emotionally as it did under placebo. And also when we look at their brain activity, we saw a reduced signal exactly in this brain region, which we expected it. So a reduced social pain signal under the influence of psilocybin. Now, we were not only interested in social rejection, we were also interested in what does it do with empathy, because of course, the world probably needs a lot more empathy. And we also know that in some patient populations, they show reduced empathy. Now, empathy is not a single construct. Empathy can be cognitive and it can be emotional. So cognitive means that I know what the other person is feeling, but that doesn't necessarily mean that I need to have the same emotions, that I need to feel the same thing. Emotional empathy, on the other hand, means that I do feel the same emotion as this other person. So we tested this here, and what we saw is that under the influence of psilocybin, participants showed more emotional empathy. They felt more strongly with other people than under placebo. They did not show differences in cognitive empathy, though. But maybe cognitive empathy is an ability that you cannot just change acutely by administering a substance. Maybe this is a process that is learned over a long time, or maybe it is something that we would see changes in maybe a day or a month after we administered the substance. This still needs to be tested. On the other hand, also seeing these no changes in cognitive empathy also is encouraging for us, because it means that our participants were able to perform exactly the same way on this test as under placebo. So they didn't show any impairments either, which means that we really can trust these effects. The other thing that we did in this study is we tested moral decision making. So do people behave more morally under the influence of psilocybin? We did not find any differences there either. Maybe it's the same thing with cognitive empathy. Maybe this is something you cannot just change by administering a substance and testing it acutely. Maybe these are things that take time, or maybe psychedelics just don't make us behave more morally. So to sum this up, we've seen that self and social cognition are processes that are intertwined. Psychedelics reduce rejection sensitivity, and at the same time, they increase emotional empathy, but they do not acutely affect moral decision making. So in the limited amount of time, I've only been able to touch on a few social processes, of course, and only on LSD and psilocybin. Of course, there's MDMA, and we know that MDMA changes a lot of how we feel about other people. So if you are interested in this topic, we've tried to sum it up in a paper published late last year. And there is a whole summary of how these different substances change different social cognitive functions. So this brings me to my last slide, really. So what do we learn from all of this about the possible mechanisms of how these substances act in the brain and how they might be able to treat psychiatric disorders? Well, one hypothesis is, of course, that they just change the serotonin to A receptor system. You remember from the beginning of my talk, they act on a very specific receptor system. Maybe they change that in any way. We have, I think, one study published which investigated this in healthy participants about a week after administrations. They didn't find any consistent effects on the serotonin to A receptor system, but maybe it is in clinical populations. We also know, mostly from animal studies, that psychedelics induce neuroplasticity. So what that means is they might facilitate learning. And that obviously might be interesting in the treatment of psychiatric illnesses, because usually these people need to learn new skills, or they need to forget things, bad things that has happened, or associations that they have, for example, between specific stimuli and cues in the case of addiction disorders, for example. So induced neuroplasticity is a very important concept, facilitated learning. However, this has only been shown in animals so far, and we're starting a study, hopefully very soon after this global corona crisis, that will tell us whether psychedelics also induce neuroplasticity in the human brain. We've already talked about alterations in brain networks, so maybe these alterations that we've seen that I've been showing you before, maybe they can normalize pathological networks that we see in our patient populations. We've already been touching a little bit on reward and emotion processing, so maybe being able to approach negative life events because our amygdala is not reacting as strongly as it usually does is something that might be very helpful in a therapeutic process. Also, a lot of patients report that they gain some sort of insight into dysfunctional behavior, which is, of course, very important in a therapeutic process. And here we come back to our last topic. Social connectedness is certainly something that we should put on the list of possible mechanisms of action. So people may be more empathetic, they may more easily connect with their social environment, also with their therapist. And I'd like to close this talk with a quote that I found in the reports from one of the addiction trials. So I was in love with the whole room, the people in it. Love is a pretty big distraction from addiction, and my attention kept going back to it, that great feeling of acceptance. And I really think this sounds like a very, very therapeutic experience. So thank you all for being here and for listening to me. And of course, I have to thank my whole team at different universities who were involved in this work. And now I'm coming back to you.
Thank you, Katrin. That was such an awesome presentation. And just for you to know, there's 1 million messages complimenting your work here. The chat has been blowing. Everybody is super happy. To me, it's hard to understand, as I said, but you totally win me over during your presentation. When more towards the end, when you're talking about these experiments about people feeling rejected, and it was pretty moving to me. I mean, the simplicity of those kinds of experiments and the human nature, the beauty, the simplicity of main things. And I was driven back to I got this job, and I used to live in the semi-desert of Mexico. And anyway, it's a long story, but there was the situation where we would all go to lunch together, and I would sit by myself, and then I would see, you know, all the colleagues passing and greeting me and going sitting on another table. And I stayed like 2 years like that, like feeling this deep rejection, and it was so painful. And it was like feeling like I was back into high school or kindergarten, like just a very small thing, that like geography of people passing and moving and sitting elsewhere. And I really felt a lot of comfort in peyote, and I started to cultivate a cactus garden. So I started to create this relationship with plants and find this kind of comfort, and I felt liberated, like science showing to me, you know, that these plants have really the ability to work on some kind of level related to all of that. And the other aspect that I found interesting in your presentation, when you say, so I could relate to the element of connectedness and isolation, but I did not. I was surprised when you said that it did, in your guys' experiments. So okay, this is a lab with, you know, German and Swiss people analyzing people's brains and stuff. I come from the world of ayahuasca and the Amazon and the jungles, and you know, also did my background in Mexico and peyote. But in the world of ayahuasca, it's very famous for ayahuasca being some kind of like antidote to lie, and that people, there's stories, there's a lot of tales that people will, for example, drink ayahuasca and then start shouting like, it was me, I stole that, or, you know, I confess, I'm sleeping with your wife, or things like that, that it really kind of confronts you to take a moral stance. So I was a little bit, you know, intrigued by that. It's not a question, it's just a comment. We have a lot of comments. I'm gonna read the questions. I just wanted to make these 2 comments, and I wanted to make one generic question that is pretty basic. And this webinar unites people that are like, they're the top geeks. You can see people like discussing all kinds of details, but there's also newcomers. So we kind of have to have that in consideration. It's a basic concept, but I don't think everybody knows that slide that I think everybody can relate. Why? And you talk about rigid patterns of thinking or ruminative thinking. And I myself, you know, check for that. And I think a lot of us as well. Can you explain what is that? And why do you think psychedelics can help with ruminative thinking, in relation to that, to depression?
Yeah. So, I mean, we see these ruminative thinking or rigid thinking patterns in a lot of disorders, mainly in depression, as well as in addiction disorders, and of course also OCD. All these illnesses are currently being tested for the treatment with psychedelics. And I think this is really something that they have in common. And what that means is that your thoughts really circle around a specific topic. In addiction, it may be the drug. In depression, it is usually about, you know, bad life events, or thinking strongly about yourself, being like, well, I'm the worst, I'm a bad partner, I'm a bad mother, etc., etc. And so usually in healthy people can usually distract themselves from these thoughts. I mean, to a certain degree, they usually affect all of us. But when it becomes pathological, you get so stuck in these thinking patterns that you cannot escape them anymore. So your whole world is revolving around these thinking patterns. And that is obviously, that's terrible, because I mean, that makes your life really, really miserable. And the way we think that psychedelics may pave a way out of these thinking patterns is when we look at the connectivity patterns in the brain, we see that the information that we get is put together in a very different way than usually. And if that happens, if even if it's just for a limited amount of time, if you integrate the information you have about yourself and you have about your environment together very differently, it kind of forces you out of these thinking patterns, because you cannot really go back to them, because the information is just integrated differently. And maybe just experiencing this this relief of these thinking patterns, even if it's just for a short amount of time, that can already be helpful to kind of know that there is a different state where you don't have this ruminative thinking patterns. And that is something that you can work on then in the integration sessions and in the therapeutic sessions after the actual experience.
Yeah, thank you. I love this idea that it's not necessarily like inventing the wheel, but it's about combining the resources or the ideas, the elements that you have in a different way.
Yes.
So that can create novelty. And just before we move more, can you just say, if you don't mind, I mean, just going to your comfort zones. Everybody do their job. I do mine, you do yours. Why did you started to want to study this? Do you have any like personal relationships to this topic, or how did it happen that you end up being on a webinar on April 16, 2020, in the midst of a global pandemic? Were you since small, like trying to look things in the lab, or tell us?
Well, believe me, if you had told me 12 years ago, I would say you're completely crazy. So no, for me, it's really, I studied psychology. I was always very interested in how the brain works. And especially I was interested in the neuropharmacology of the brain. And that means like what has to happen on a chemical level, if you think back to these synapses I showed, right? What has to happen there to make us like feel this emotion, or make us do this, or make us choose, salad over bread, for example, right? All of this kind of has an origin within our brain, within our synapses. So I was interested in, because that was the thing that I was really like, I just can't understand. And so I went on to do a PhD. When I did that, I wasn't even aware that something like science on psychedelics even existed, right? So I did my PhD on addiction disorders and long-term consequences of chronic cocaine use mostly. And I love that work, and I think it is very, very important to make the treatments available for addiction disorders better. So I thought it was very important work, but it didn't really answer the questions I had about, why is the brain working like it does. And at that time, I got to know Franz Vollenweider and his group, and I thought, wow, hey, that I didn't know that this type of research even exists. And so I started to learn about it, and I thought it was fascinating. And I thought like, well, this is really the window into the brain that I was looking for. And after I finished my PhD, I did a postdoc in this group, and and I never stopped working with psychedelics since then.
Okay, and can you share like what is it to be a woman in doing scientific research about the brain, and how is it your work environment and all those things?
Yeah, I mean, it is, especially neuroscience, of course, is it is very much dominated by men, of course. So my experience is that we certainly need more women in this field. We need more of a gender balance, because what my impression is that, these balanced teams just really work better together. And I'm so proud now that I have my own group, that I have 3 highly talented female PhD students, and we're slowly increasing or balancing the ratio, the gender, or moving towards a gender balance there. But it needs people, we need to educate the women in science, and we need to kind of motivate them and showing them a path towards a career in science. And well, I'm hoping that I can be a part of that, and I think, you know, being here probably hopefully already motivates a few of them to consider this as a career path.
You are a part of it, and I think, you know, it's exactly like that, by having examples and by speaking up about this topic. And I want to remind that since we're, you know, in Switzerland-based territory, Annie Oak and Maria Mangini like to tell the story on how a few representatives went to the 100th anniversary of Albert Hoffman in Basel, I think 2007 perhaps.
Mm-hmm.
And I think of like 90 speakers, they had like 6 women or so, something like that. That's the story they tell. And then there's like pioneering figures. They came back to the US, they were upset, and they decided to create the Women's Visionary Congress, and they have been stimulating the presence of women in research. And since then, I think a lot has evolved. I also have a affiliation besides MAPS to Chacruna. We also promote the voices of women. So I think definitely. I also wanted to ask a question about, so you have one foot in Yale and one foot in Zurich, and the US is, as you know, is pretty US centric, and Americans frequently don't look much beyond the US. And I, as a foreigner, I also have more like global, you know, cosmopolitan vision. What are your impressions? I don't know if you can say anything about this, but like on how the field of psychedelic science is developing in the US versus Europe nowadays. Do you have any comments about this?
It's a bit hard to tell, because these are, I mean, we're living in exciting times, right? A lot more labs are getting interested in doing this research, and it seems that some countries are opening up to the idea of having people do this research. For me, it's always been, well, Switzerland has always been for me the place to do it, because well, obviously LSD was discovered here, or the effects were discovered here, and that kind of has led to a long tradition of research on psychedelics in Switzerland. And that being said, that means the research here has never really stopped. So at the institution where I work, we're looking back at I think 25 years of experience with these types of studies, and that, of course, is much longer than any of the other modern research centers, be it in the US or anywhere else in the world. So I'm very happy and very grateful working there. But of course, also in the US, a lot of very talented scientists have begun to do this work in the last about 15 years, yes. And there are so many exciting results coming from basically all over the world right now that I'm very excited to have the opportunity to work in both places, to see how things are developing in the US as well as in Europe. Of course, we still need to be aware that you cannot just, run a study, give a psychedelic to someone, and then hope that everything will be fine, right? So these are highly psychoactive substances. We need to prepare people, we need to take care of people when we do these types of studies. And these are not studies that you rush, right? They need time. You need time for your participants. You need time to prepare them. And so I really hope that everyone who's interested and who's now entering the field and hopefully, showing or uncovering a lot of exciting results, that they are aware of this, and that everyone is doing the research rigorously and taking very much care of their participants. And so my recommendation is like, well, if you're starting to do this research, this is nothing you can rush in any way.
Yeah, thank you for saying that. Definitely, it's a suggestion of the panacea that we're gonna solve all the problems. So as much as I would love to continue a million questions to you. I'm gonna read here some of the selected questions we got. So Jacqueline is saying, I'm excited. I recently been accepted into PhD program starting this fall and plan to continue on a neuroscience trajectory. I would ultimately likely to pursue a career in psychedelic research, and the programs I have been accepted do not directly offer this. I would like to ask you if you could kindly suggest a graduate research focus that would lead into a smooth transition into the field.
Yeah, I mean, it really, so there are many paths to doing research on psychedelics. I think the most promising is to become a good researcher, become a rigorous researcher, know what you're doing, know your methods. And then it really depends on what exactly your questions are with regard to psychedelics. So if you're more interested in basic pharmacology, for example, then you should probably work in an animal facility and do your tests there. If you're more interested in what's happening in the human brain, then neuroimaging is probably the way to go. On the other hand, if you're more on the therapeutic side, then some kind of clinical fellowship might be the right thing. I mean, even a lot of philosophers are very interested in psychedelics. So there are many ways which can lead you to doing this research. And I think the most important thing is to know the questions you have, and those will show you the way of learning what you need to learn to be able to answer them at one point.
Yeah, cool. I get a lot of questions like that myself, and I like to give people an exercise. I have a nickname called Tia Bia, Aunt Bia. So I like, when you have an idea and you don't know what to do about it, like write a paragraph. It's a very basic resource, like trying to flesh out an idea of what you want to do. A paragraph, a title, 250 words, it can really do wonders, because it does help you start narrowing down, different possibilities. I want to jump to the question by Jan. Hi, could Catherine please comment on the serotonin syndrome in association with the use of SSRI medication? Which would you recommend that people do not take SSRI or any other antidepressants prior to the use of psilocybin or LSD to prevent serotonin syndrome?
Yeah, so I wish I had an answer, because we honestly, we don't know. There have not been any formal studies on that. And since we just don't know, except for anecdotal reports, the anecdotal reports say that, well, probably not. However, since we don't have any scientific evidence for that, I will rather recommend to be cautious until we have these studies. And at one point we will have them, just not right now.
Cool. Yeah, I also get questions about this, about ayahuasca. I think there has been a paper published warning people not to mix ayahuasca and antidepressants, but there's not real evidence. It's more like a theoretical risk, but on the prevention side, people have chosen that way. In the Brazilian ayahuasca field, there has been a lot of anecdotal evidence that people have combined, and goes by doses and kilos of the person and specific stories. It's difficult to do like a generalization. We have a paper in our book published in Germany by Lit Verlag that talks about epidemiological overview of the UDV. They did a survey and tried to make see this in large populations, but there's not a lot of real data, as you say. So I think that on the prevention side, we do recommend people not to, because if we say, well, there's not evidence, then maybe people will say, well, okay, we should just do it. And
yeah,
I think caution is good on this regard. There's another question that is talking about interaction of psychedelics and cannabis. There's what could you talk about that?
in interaction, in which way? On the subjective effects?
Between serotogenic psychedelics and, can you comment on the brain interaction between serotogenic psychedelics and cannabis?
Well, unfortunately, I don't think I can, because I don't think there has been any study on that. So cannabis is acting on a different system in the brain, but I am not aware that the interaction of both systems has been studied in any way. Maybe that is something very interesting to do in the future. However, as with other medication as well, people are always careful. Mixing two substances can have unpredictable consequences. Therefore, these types of studies are actually really hard to do. But it would be terribly interesting. I completely agree.
That's a funny way to put terribly. It would be terribly interesting. So people looking for ideas, you got an idea here. For example, given the difference in receptors between LSD and psilocybin, does that mean that they would be suited to different mental health issues?
Mm-hmm. Very interesting question. Also not one that is easy to answer. So what I can tell right now is that we have blocked specific receptors and then looked at which type of receptor is really is responsible for the effects of psychedelics. And there we've seen a common pathway that basically all serotonergic psychedelics act mainly through the serotonin 2A receptors. And most of the other receptors don't seem to be that important. Again, we're still kind of, as I said, like this new area of research where we can actually have the methods to test things like that. It's still kind of new. And we're still learning a lot. So it might eventually turn out that, for example, other specific receptors are important too, but right now all we know is that probably the serotonin 2A is the most important, and that's the same for psilocybin as well as LSD. So for the time being, I would say, well, probably not. But then again, we know way too little about what exactly it is that is making people feel better. And that is exactly why I'm saying that we need all this research. We need to be able to understand what it is that is people making better eventually. And then maybe at that point, we can answer these questions.
Thank you. Another question by Asdrubal and Alex. What is your opinion on the role of psilocybin and LSD on neuroplasticity, pruning and resprouting synapses, and its therapeutic implications?
Mm-hmm. I personally think that this is incredibly exciting. So I think if psychedelics indeed open a neuroplastic window, this could be clinically very, very useful, because then we can use this window to learn new things, to maybe help people work through things that they eventually kind of want to not necessarily forget, but be less traumatic, for example. So I think there is a massive potential behind induced neuroplasticity. However, so far this has only been shown in animals, and we need to test whether this also holds true for humans. And this is actually a study we are hoping to start soon, where we will test whether we can see this induced neuroplasticity also in humans, and if yes, whether we can leverage this to make the therapy even more effective.
Cool. There's a lot of questions asking for references and papers. So I just want to tell everybody, in our site MAPS, if you go to the MAP site, right on top, there is the webinar. You click there and there's all the sessions. We offer a series of resources that are recommended for people to further delve and research. So go to our site, and Catherine has has given already a few resources over there. And I wonder if you can, Jill asks, can you review briefly the dosages of psilocybin used in your research?
Yeah. So with psilocybin, we mostly dose per weight. So that means most of the studies have used something around 0.2 milligram per kilogram. So we're talking about somewhere between, well, 15 and 18 milligrams. With LSD, we've used 100 micrograms, except for the microdosing study, which was 13 micrograms.
Katrin, there's so many questions. I am overwhelmed. I just want to tell you that, you know, this is a potential good career path for you to keep on giving a lot of lectures, because there's a lot of questions. I hope that this is the first of a series of appearances for you. You have sparkled people's minds. There's like 154 questions. Brad is selecting them, passing them to me. I want to say to everybody, I'm doing my best, but there's a lot of questions, so maybe yours don't get picked. Teresa and Yvonne wants to know, can you say anything about the effects on psychedelics on developing children? On children, adolescents' brains?
Yeah, we don't know. We just don't know. I mean, we're so happy that, as I said before, it's been in Switzerland, it's been about 25 years that we are now able to do these studies again. Obviously, we have a lot of data from the 50s and 60s and early 70s, but after that, we had this break, and now we're kind of slowly starting to do this research again. So obviously we've been restricted in what exactly we can study, and we're very happy that we're even able to do what we're doing. And now we've finally reached a point where more countries are allowing this type of research, and we haven't been able to answer all the questions. And doing research in kids or young adults is terribly hard to get ethical approval for. And there's obviously also a reason for this, because you don't want to jeopardize someone's future for one single experiment, right? So all I can say is, no, we don't have any data on that at all. But with, you know, increasing research in on the topic, and maybe even medical use, I'm pretty sure that at one point we will get this information.
Yeah, I want to say that, you know, I'm not a scientist, a neuropsychologist, I'm an anthropologist, and from the training I have and the background that I have, what you can say about traditional people is that frequently, you know, in shamanic context, children and teenagers do use psychedelics, and it's common, it's part of the life, and, you know, of integrated into culture and society. We, even in the MAPS bulletin that I worked on, Women and Psychedelics, you can find all our bulletins online, also on our site. There's an opening article by anthropologist Steffi Schaffer talking about how Wichol or Wirarika women breastfeed their children when they're eating peyote. They consider it's important for the children to grow strong. It's a beautiful article. Also, there's you know, a lot of literature on how children and teenagers are given ayahuasca, and that's part of their training. Or even sometimes, you know, if they're not behaving well, the mother will take them to a healer and say, like, now, you know, you sit here and you meditate a bit. It's also part of the ayahuasca religion. So, you know, there's different angles to how we study these things. I think it's definitely one of the largest taboos, no, because it's like how mother and pregnancy as well. In some, there's a lot of different approaches to this question. There is a question about schizophrenia by Nick. There have been risks associated with treating psychotic patients, such as schizophrenics, because the psychedelics may push them over the edge. Have any studies supported or rejected this theory?
Yeah. So, well, again, the answer is we don't really know. We've seen from again, the old studies in the 50s and 60s, that it doesn't seem to be particularly helpful for schizophrenia patients. Then again, the definition of schizophrenia was very different back then, and also the therapeutic methods were different. So that is kind of why we can't translate a lot of results from back then to today, but not necessarily all of them. And what we don't, so I mean, we have this, or we're pretty sure that giving psychedelics to schizophrenic patients is probably not making them any better. Whether it can cause psychosis, and with that I mean long-lasting psychosis, so basically schizophrenia, is an unanswered question, really. But then again, this is also again very hard to tell, because obviously we cannot give psychedelics to at-risk populations and then just see whether, you know, they get psychotic or not, right? So we have to be careful, and we don't recommend anyone who is at risk of developing psychosis to take these substances. And again, here, same thing with kids or other at-risk people. Same thing, for example, with people with heart problems. Rather stay on the safe side. And maybe at one point we will have data that show otherwise, but as long as we don't, I can only recommend staying on the safe side.
Yeah, that's very interesting also, because some of the experiments that were done back in the day, they didn't necessarily have ethical standards that we do have today. So my colleague, Dr. Monica Williams, was presenting on, we co-edited a special journal edition about equity, access, and diversity in the field of psychedelic medicine. And she's doing a historical research about experiments that were done, like with prisoners and LSD. And so like the contrast, like African-American prisoners being given sometimes LSD without any kind of information or preparation, and comparing to like white, wealthy subjects that were not prisoners and were getting preparation and integration and all of that. And then the kinds of results and the kinds of data that is also produced in this research, it's complicated. Like you really need to take a lot of things into consideration when moving on experiments. And then the whole dark period of the CIA investigating the potential of the substances, like to do mind control and to improve investigation techniques. And we also know, yeah, all these other kinds of American Psychology Association has been involved in scandals on doing research on how to improve their methods for torture. And there's a sketchy area. And yeah, psychedelics are fascinating because of that, because we're dealing with borders and with pushing borders, but this pushing borders can also go wrong. I want to pass to the next question. So there's a classic, a lot of people are asking between differences and similarities between the functional effects of microdoses versus normal psychedelic doses, which is interesting also how you call it, because one is micro, the other is normal, or is it macro? Like, how do you even call this?
Yeah, I think we know so little about microdosing, and we cannot even agree on the right terms, because some people also already call it mini dosing, for example, if it's above 10 micrograms. So, I mean, what we know is that it's basically, I mean, well, to be very honest, we know very, very little about it. And pharmacology is terribly complicated. So there are some substances which basically just have weak effects in a low dose and strong effects in a high dose. There are others which kind of have what we call an inverted U shape, so strong effects on lower dose, then, you know, it's something in the middle, and then it goes up again. So pharmacology is very complicated. For LSD, it seems more like, well, weak effects on a very low dose and high effects on, or strong effects on a higher dose. But really, we only have very few placebo-controlled studies with a so-called microdose. I think it's like 3 or 4 by now. And what they have shown on a behavioral level is more or less nothing. So no change in creativity, if you can even measure that. No change in concentration, maybe a little bit of a lift in mood, but that's basically it. Again, then on the brain side, we have seen changes that might indeed be interesting. So for me, the big question with microdosing is really, is microdosing, or is LSD in general, a substance that can acutely make healthy people even better? So that is usually the question when people talk about microdosing. Is it like a cognitive enhancer? For the time being, it looks like not necessarily. But then again, it might make people better who have some kind of difficulties in emotion regulation, for example, depressive symptoms, etc. But we really cannot answer the question quite yet, because we just have, again, maybe 4 studies out there which tested a small dose of LSD.
Thank you. I continue to be overwhelmed with the amount of questions. Yes, please drink water. There's a few more. Officially, we should end at 1:30, but we're gonna give 5 minutes extra because of the people's claims. But let's, no more questions, please, because we don't have any more time. Has there been studies conducted on the long-term effects on brain connectivity? Do any of these changes last beyond the immediate effects of the substance? What about HPPD?
Mm-hmm. So there is unfortunately only one single study who has looked at long-term effects on brain connectivity. And that was only published about a month ago by the wonderful scientists at Johns Hopkins. And what they have found is that some effects persist after the acute effects, especially 1 week after administration. However, about 1 month after administration, things seem to go back to normal. That is unfortunately the only study I know of right now that has tested that. Again, hopefully more studies to come. And the second question, HPPD, well, it seems to be rare. We have not had a single case in our studies, and we've tested, I think, by now probably about 1,000 people. But, and here comes the big but again, here we are in a research setting. We have patients which were selected based on, they needed to be healthy, etc. They needed, they don't have any relatives with any psychiatric disorder, well, not necessarily psychiatric, but they don't have people with psychosis disorders in their first degree relatives, for example. And they obviously are in a setting with trained people. And so it is a very different setting from, for example, taking a high dose of LSD at a festival. So that doesn't, so what I'm trying to say is, I'm not saying that HPPD cannot happen. We don't know why it happens, and we don't necessarily know when it happens. But based on our research, we think that it might have something to do with the dose, with the setting, and probably with, whatever you bring to this. So if you have, again, if you have people in your first degree relatives who have schizophrenia, or if you know that you have a tendency towards psychotic experiences, it might just not be a good idea, because theoretically it is conceivable that those people are more strongly affected by HPPD than others. But again, since it's so rare, it's really hard to do research on this phenomenon.
Catherine, do you have energy for a little bit more? 5 minutes?
We'll do 5 more minutes.
Okay. All right. Trying to get the most out of our presenters, but really going to the end. So one question is about, can you say something about your understanding of the action of psychedelics in interrupting the process of addiction?
So basically, in the mechanism of action in addiction, if I understand correctly.
Yes.
Yes. Again, oh my God, it's so many, so interesting questions. And I feel like for most of them, I just have to say we don't know. Because basically the last slide I showed, these are all hypotheses. Most of the studies that have been done so far in clinical populations have not looked at any mechanisms except for, well, maybe correlations between mystical type experiences and clinical efficacy. So we really don't know right now, but all the things that I've listed on this last slide may have an impact on how these substances kind of may break the circle of addiction. And here again, I mean, I'm saying about neuroplasticity, like maybe loosening the connections between associations with the drug, reconnecting to the social environment, insight into dysfunctional behavior. All these things may have a very positive influence on people's life with addiction.
Yeah, that's definitely a huge topic, and I think we should make a whole webinar about brain and addiction. So the last very question is, can you talk something, why the characteristics of some people brains that make them more sensitive to the psychedelic effects? So why do different people react differently? Is it in the brain?
Well, again, we don't know. Certainly the brain is certainly part of it. It's probably not the whole story, but I think it is an important part of it. So when you look at predictions of the effects, it's really hard to find anything that is really predicting the experience. Also because it just varies so much within an individual from experience to experience, right? You can have wonderful experience one day and a terrible one the next day. So so it is very hard. What we've seen is that people who are in general very anxious, they often don't have a good experience. Also people who have a high need for control are maybe have a more difficult experience than others. And then again, this doesn't necessarily hold true for everyone. This is just like a general statement that we've seen in the research. Then again, so much may depend on really the setting, how people are prepared, etc., that it is terribly hard to predict. But we are working on that, because now that we're kind of slowly moving into the clinical area, I think these questions become more and more important, because at one point it would be great if we can predict in a way whether people will react positively in a treatment setting to these substances. So we are working on it, and we're trying to find out if there is a pattern in the brain that can explain the variability of responses to the drug.
Fantastic. This is the time, if we were on a stage where everybody would clap and stand up. So we're, virtually clapping. We are going to conclude. I know our presenter is from Germany. I lived in Germany, and one of the things that I most enjoyed about that country was punctuality. The fact that when you schedule a lecture, everybody shows up and starts on time and finishes on time, and people don't interrupt each other. Some of the very fascinating aspects for me of Germany as a Brazilian, and I very much appreciate punctuality. So thanks for extending a little. I just want to give some final announcements, and thank again you on behalf of Brad, myself, and all our team, and wish you much success in your young woman and doing fabulous research, and I hope that you continue to rise and shine, and it has been a wealth of knowledge. And yeah, I just want to know if people want to get in touch with you or support your work somehow. Do you want to refer to a site or email or any kinds of supports that you might need?
Absolutely, anytime. So it's easiest to reach, it's easiest to reach me on my email address, which is greller@eli.uch.ch. That is kind of complicated in American because one is like the Z and the other one is the C. But you will find me online. So if you Google me, you'll certainly find me. You'll find the Institute's websites, you'll find the email addresses. I'm on Twitter as well. So however you want, just reach out to me anytime.
Well, thank you, Dr. Preller. I want to give, an announcement of a few things that we have. So it's a vibrant spring and we are on quarantine. There's a global pandemic, but life goes on. I want to send everybody a lot of love and wish that you are all stay home, please, including Brazil that have that very, very tense situation right now. Ask people to stay home, stay safe, take care of yourselves, focus on your mental health, stay together and united. We very much need to be on our bests at this moment. I want to invite everybody for our next section, which will be next Thursday, April 23, about decriminalization, same time and same place. If you want to give us feedback or if you have important things to say, I am also available to talk to everybody. My email is there, bia@maps.org. I'm also available on Twitter and Instagram, and I'm putting it right here at @labatebia. But more than that, I have some cool announcements of some hot stuff that is right on the oven, just about to happen. So this weekend, dear friends, firsthand, without anybody knowing because we didn't announce it yet, we're going to have a super nice, MAPS will be announcing very soon. So subscribe to our email. I'm also going to put it right here for you all to see, MAPS with an update. Well, that's the parenthesis was a mistake. Brad, help me out. Put this email over there. We're gonna have sorry about that. On Sunday, April 19, MAPS is hosting a free virtual event called Bicycle Night to celebrate the 77th anniversary of psychedelic chemist Albert Hofmann's first intentional experience with LSD in 1943. We will be announcing more details about this event soon, so subscribe to MAPS emails or follow MAPS in our social media if you'd like to learn more. So that's at night from 5 p.m. to 10 p.m. Before that, we're going to have, MAPS is also proud to be a co-host of a very awesome event. So this Sunday, April 19th, we're going to join the Society for Psychedelic Outreach, Reform and Education, SPORE, from 8:45 to 5 on a day called Sacred Solidarity. That's a free online festival celebrating reciprocity and the power of psychedelics to help heal our relationship with Earth and each other. My dog is excited about the event. I'm sorry about that. MAPS is a proud co-host of Sacred Solidarity, which includes sessions featuring mycologist Paul Stamets, visionary artist Alex Gray, Rick Doblin, Gabor Mate, Ismael from MAPS, myself, and a bunch of other people. If you want to participate, it's free. All the proceeds are going to donating to COVID. We accept donations. Proceedings are going to help COVID workers. I want to also tell everybody that we are organizing a conference called Psychedelic Liberty Summit. It's on April 25 and 26, and you can join the conference by clicking our site, chacruna.net. That's not so this weekend there's Bicycle Day and Bicycle Night. Next weekend there's Psychedelic Liberty Summit. May 2nd and 3rd we have Awaken Futures and much more to come. Thank you, everybody. Thank you, Catherine. Have a nice week and see you next time. Thanks for the team, Bryce, Brett, and everybody. Big hug from San Francisco. Bye-bye.